When we think of electricity, we usually associate it with man-made objects like batteries, mobile phones, computers, etc. But electricity is also a fundamental component of almost every biological organism, including us humans. Every cell in your body uses electricity for a variety of reasons, such as cell communication, cell division, and cell development. But what happens when these electrical patterns go wrong? In this video, we'll explore how cancers are electrically different from normal cells, and how recent technologies may even make use of this fascinating form of bioelectricity. Our cells don't use electricity in the same way that electrical devices would. Every cell has a membrane which separates the inside of the cell from the outside. This membrane is semi-permeable, meaning that it controls what enters and exits a cell through tiny channels that move ions in and out. As a refresher, an ion is a positively or negatively charged atom. So when ions enter a cell, they can either make the cell more positively charged or more negatively charged. This movement of ions changes the voltage across the membrane. Like in devices, this voltage controls signals, communication, and responses. In the case of our cells, the default state is for the inside to be negatively charged compared to the outside. This is called resting membrane potential, and while the exact value of the resting membrane potential depends on the cell type, it is still vital for many cell functions. When talking about the membrane potentials, scientists use the fancy term depolarization, which simply means the membrane's voltage has gone up. In other words, the inside of the cell is more positively charged than normal. In normal cells, depolarization acts as a signal to start processes like growth and development. But this is also where cancer comes into the picture. The main premise behind cancer is uncontrolled growth and division of cells. Interestingly, compared to normal cells, cancer cells have been shown to be chronically depolarized, meaning that their resting membrane potential is unusually high at all times. This understandably causes their high levels of growth and division. The idea that cancer cells exhibit differences in electrical patterns isn't new. Research from back in 1971 showed that depolarization was directly correlated with an increase in cell growth and division. Throughout the 1990s, more and more research showed how ion channels inside cells were very much related to cancer development, creating the term oncochannels, channels that were abnormally altered in cancer cells. A good example of this is voltage-gated sodium channels in breast cancer. In normal conditions, these sodium channels are very low in number, but in the case of breast cancer, there is an abnormally large number of these sodium channels. More sodium channels means more sodium ions going into the cell. These sodium ions are positively charged, meaning that the inside of the cell becomes more positively charged or more depolarized. As such, it plays a major role in the spread of breast cancer. One potential way to counter this depolarization is through ion channel blockers, drugs that inhibit specific ion channels to restore electrical balance in cancer cells and slow their growth or spread. While none are yet FDA approved for cancer, research in this area is advancing. Several candidates have shown strong preclinical results, such as chlorotoxin for glioblastoma and estemizole targeting calcium ion channels. In fact, one such calcium channel inhibitor, SORC13, has become the first ion channel blocker to reach human cancer cells and has shown very promising results. Ion channel blockers are not the only way in which cancer cells can be targeted by means of bioelectricity. TT fields, which stand for tumor treating fields, are another ingenious treatment that focus on cell division, otherwise known as mitosis. Mitosis is a process that makes two identical daughter cells by separating and sharing the parent cell's chromosomes. TT fields target the cellular components involved in chromosomal separation, such as the mitotic spindle. In its essence, TT fields are low-intensity, alternating electric fields, meaning that the electric field changes direction rapidly at a very specific frequency. This frequency is super important and can be changed depending on what needs to be targeted or what cancer is involved. Since TT fields usually target mitotic spindles, which are smaller organelles, they are tuned to a frequency that is too weak to target larger organelles, but just right for smaller organelles, like the mitotic spindles. Mitotic spindles are tubulin dimers, which have an electric dipole and are very susceptible to electric fields. When the TT fields are set, they interfere with the process of mitosis, and these effects can lead to cell death. Interestingly, TT fields can equally affect healthy cells dividing as well. However, because cancer cells divide at a much faster rate than healthy cells, which otherwise divide very slowly, cancer cells are affected far more than healthy cells. TT fields are already FDA approved for glioblastomas, which are aggressive and common brain tumors. To use TT fields, electrodes are placed on the patient's head and they are used to generate an electric field. TT fields are continuous treatments, meaning patients who undergo TT fields must wear them continuously for 18 hours a day. Although TT fields are not a definite cure to glioblastoma, they can extend survival and improve quality of life. TT fields are also being developed for other cancers apart from glioblastoma and are showing exciting progress on that front.